in a response. And you'll see examples of this soon. So if it sounds abstract, it will get a little bit more clear or specific later. And so you can have two types of receptors, and they're dependent on the type of signal you can have. You can have this. You can have lipophilic signals. By the way, lipophilic means non-polar essentially. Lipo means lipid, philic means love or like. So lipid loving is non-polar. So if it's lipophilic, it doesn't need to worry about being transported through the membrane or anything like that. It can just diffuse the membrane, remember, because the membrane is mostly nonpolar. And so these molecules can simply go straight through the membrane. They don't have to have any special system to bring them in. And once they go in, they can then either bind into some kind of receptor in the cytoplasm, or they can go directly into the nucleus right here and bind to some protein in the nucleus, or bind somewhere on the nuclear envelope, nuclear membrane, and then create some change. These responses tend to activate some kind of protein synthesis. And so that, once again, requires transcription, translation, the forming of protein structure. So because of that, since it involves protein synthesis, these processes tend to take a long time. These are very slow responses because they require production of protein. Now, you can have lipophobic, or you can call it hydrophilic, same thing. You can have hydrophilic, so polar molecules, or molecules that just can't diffuse through the membrane, cell membrane, and so they're just going to meet up with a membrane protein, so some kind of membrane receptor. And when this happens, it's going to create some kind of intracellular reaction, which will then lead to response. And because it doesn't necessarily involve the synthesis of proteins, it happens a lot faster. It happens very quickly, within seconds, within minutes, rather than within hours. And so once that ligand hits the receptor, this is the basic process that happens. So the signaling molecule is always considered the first messenger. It binds into a receptor which we call the transducer. And this transducer then will activate these secondary or second messengers. These are the messengers that will go tell other proteins, other structures in the cell to do stuff. And so they'll go to targets, and then they'll create a response here. And so a lot of times in these hormones and whatnot, the hormone actually doesn't really get enveloped into the system, and it doesn't propagate into the system and then create all this change. It actually requires other agents, other proteins, these secondary messengers, to go spread the word. And because of this, because you have these secondary messengers, you don't need you don't need a high concentration of first messengers. And you'll see why in a bit. Just hold on to that thought. And so here is a graph. So here, this is what I just showed you earlier. So this plot right here shows you all the possibilities of things that can happen. This shows you a whole bunch of things, and it looks really complicated, but I'll let you know that not everything on the figure happens every single time a signaling molecule binds into a membrane receptor. A subset of these things will happen. So for example, this is the first messenger. This will bind to the membrane receptor. This will initiate the signal transduction. So when we talk about signal transduction, that's the process of taking the first messenger and creating change intracellularly. Basically activating mechanism inside the cell based on something on the outside. So we're transducing the signal is basically what it is. And so the transduction can require something called amplifier enzymes that will amplify the signal. which will then lead to the secondary messenger molecules. And a couple things can happen. Well, it can then open an ion channel, and this ion channel can then release a whole bunch of intracellular calcium, for instance. And that calcium then can go affect proteins that can create a response, or these second messengers can then affect other proteins, which will then affect other proteins to create a response. And so there's a lot of different paths. And I'm not going to explain every single path. We're going to give you a couple examples today, and then if you ever need to learn a new one, it'll be pretty similar to what we talked about in class. But a couple of things before we get to those couple examples. First, every signal transduction takes in shape of some kind of cascade. A cascade is basically when a signal activates one protein, and that protein can activate another protein, and that protein activates another protein, so on and so forth, until you get to the actual activation that gives you the response. So if in this example, turn some kind of substrate into a product here. So you're activating some kind of like enzyme. And so by itself, this seems kind of stupid, right? Like why did I have to go activate B if this could have just done this, right? And so the answer is yes, it's pretty stupid. And if this was the only thing that was happening, it'd be pretty silly. Like this would not really serve much purpose as far as efficiency goes, but it happens because it is coupled with phenomena of amplification. And so here once again we have the first messenger binding to the receptor, and this receptor activates this amplifier enzyme. That's what this AE stands for. Amplifier enzyme. And this amplifier enzyme will create secondary messengers, which will then in turn activate even more messengers, and then those messengers will activate even more messengers. And so this is where we have this amplification event, where one molecule gave rise to a lot of messengers. So this process here is a cascade. You could say like, well, this is protein A, and protein A activates protein B, and protein B activates protein C. And so this in itself is a cascade. And so of course we don't see a cascade where one protein is activating another protein and that single protein is activating another one. That would be silly. Here we're actually having an amplification. So every molecule, every protein activates a handful of molecules. Has anyone seen the movie Paying It Forward? Mm-hmm. Okay, one person. Well, for the people who haven't seen Paying It Forward, I'll spoil the movie for you. The idea is, and you might have heard the concept already, someone does something nice for you, and you're like, oh, how can I pay you back? And the person says, well, do 3 nice things for someone else. And so you got to do that. And so those 3 people, when they ask you what they can do for you, you tell them, pay it forward, do 3 nice things for someone else. And so that's the amplification effect. Supposedly, you have a world where everyone's doing nice things for each other, which doesn't really happen, but that was the point of the movie. And so that's the same thing's happening here. You're having the application effect where one protein is affecting multiple proteins, and those multiple proteins are affecting even more proteins. And so that's also why a very small concentration of first messengers can make a large effect, right? When we're dosing people with drugs or with hormone therapy, it's in the milligrams, right? Despite you being kilograms of weight, you only need milligrams of some kind of drug or hormone or whatever. And this is part of the reason why. It's because you only need one molecule to make this much of a change. It's a huge change. And this is why you can have this huge response from such a little amount of first messenger. All right. So we talked a little bit about signal transduction. Let's talk about the receptors that actually receive that first messenger. And so you have 4 types. You have the first one is the channel protein receptor. So the idea is the signaling molecule will bind to the channel, and the channel will open, it'll close, it'll do something, and it will change the permeability of a certain molecule. So if this was an ion channel, it opens, it would lead a whole bunch of ions in or out of the cell. Or if this was, let's say, a glucose transporter, if it binds, it can turn on the glucose transporter to have the cell start taking in glucose, or turn it off so it stops taking glucose. These are examples of a channel receptor. Then you have a very, very prominent receptor, a G protein coupled receptor. That's a mouthful. Oftentimes it's just abbreviated as GP04. This is a big receptor. This is a very prominent receptor, and a lot of signal transduction will use some kind of G protein complex. We'll talk about this in more detail in a bit. But this is a big one. And the key thing here is this receptor is a transmembrane receptor, because the structure goes in and out of the membrane. 7 times to be exact. And then the other two, less important or less prominent, but they're still there. You have the receptor enzymes. That's basically when this receptor gets bound, it activates or inhibits some enzyme on the inside. And then you have these integrin receptors that do something similar, but they involve the cytoskeleton. They're all about affecting the cytoskeleton. So that you'll see integrin receptors a lot in the immune system, or cells that want to move. And both the receptor and the integrin receptors are considered catalytic receptors because they involve some kind of enzymatic mechanism in their signal reception process. All right, so let's talk about these receptors in more detail. So here is the this is the channel receptor.